Biochemistry notes how biomolecules are built, broken down, and why they are important for life. First of all, biomolecules are different from other molecules found in our universe because they are only found inside of cells. They are built and used inside of cells naturally, and they can't be made naturally outside of cells. And the commonality of the 4 biomolecules is that they're all carbon-based. And carbon-based means organic, and so therefore when we hear that word organic, that's what that means. So if something's organic, that means it's carbon-based, that means it comes from life, and there's no inorganic substances, non-life molecules that have been added to it, like for example, pesticides or herbicides. Carbon is an atom that is unique in that it has 4 valence electrons, and in its outer shell, as you can see here, C6, so it has 2 in its first shell, and then it has 4 in its outer shell. And to be stable, it needs to have 4 more to make 8 in its outer shell, like you learned in chemistry. And so therefore, carbon is going to want to form 4 covalent bonds with either itself, other carbon atoms, or with other atoms. So you can see, for example, in this molecule right here, this is methane, and it's carbon that's bonded to 4 hydrogen atoms. Hydrogen atoms each have 1 valence electron, and they want to have 2 in their outer shell in order to be stable. And so if I were to show you like on the dot cam, here's carbon, and here's its 4 valence electrons. And let's say I want to have a hydrogen here that it bonds with. Hydrogen has 1 valence electron. And so they will share these 2 electrons, and now hydrogen has 2 and carbon has 1 more than it had before. And it can do that with 4 hydrogens, and therefore it now has 2, 4, 6, 8 electrons, and it is stable, and it is said to be methane, which is this molecule right here. Now, when you have 2 electrons shared between 2 atoms, that is called a covalent bond, and that's going to be symbolized by this solid line right here. So CH4 is going to look like that. Now, if it has, for example, carbon bonded to another carbon, so we have C, 2, 3, 4, and then C, 1, 2, 3, 4. If these 2 carbons are sharing their valence electrons, here's that bond right here, and then they would have to be attached to, each be attached to, covalently bonded to, 3 other hydrogens in order to be completely stable, because just remember, carbon always forms 4 bonds. So this is ethane right here, and it has 2 carbons and 1, 2, 3, 4, 5, 6 hydrogens, so C2H6. Now, if I were to have carbon form a double bond here, carbon always forms 4 bonds, so therefore you would need 1 less hydrogen for it to bond with. So therefore this would be C2H4, and therefore this would be a double bond here. When you see structures that look like this, like what is this? Well, every time there is a bend in the chain, that implies that that is a carbon there with associated hydrogens attached. So there'd be a carbon here, and there's it's bonded to this carbon here, and it's bonded to this carbon here. So that means it would have 2 more bonds, so there would be 2 hydrogens right here. So to show you that on the paper, so on the outside we have CH3, so CH3, and then it's bonded to that first carbon right here. And it would have 2 hydrogens coming out of it, and then it's going to be bonded to this carbon right here, which also has 2 hydrogens coming out of it. And then there's a double bond between these 2 carbons. That's what that represents, so double bond. Okay, and then that carbon that's double bonded is then single bonded to another carbon that has 3 hydrogens associated with it. Okay, so now let's look at this carbon right here. It has 1, 2, 3 bonds, so it just needs one more hydrogen. So this is where I messed up right here. This carbon has 1, 2, 3, 4, 5 bonds. That's not possible, so I have to get rid of one of the hydrogens. And so now you could say, what is the molecular formula? You would say 1, 2, 3, 4, 5, C5, H 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. H10. And so anytime there is a bend in the chain or in the ring, sometimes it'll just look like this. You assume it's carbon in organic chemistry. So this would be 1, 2, 3, 4, 5, 6. And if there's no other atoms, you assume that all the atoms coming off of the carbons are hydrogens needed to make the molecule stable. So the 4 types of biomolecules are carbohydrates, proteins, nucleic acids, and lipids. And they are usually very large molecules built from smaller repeating molecules. And those repeating molecules are called monomers. Monomer means one unit, so mono one, mer unit. And when you bond a bunch of monomers together covalently, that forms what is called a polymer. And so inside of cells, tissues, organs, and organ systems across all life, doesn't matter what kind of organism you are, you're mostly water, around 70% water, and then the other percentage is going to be these 4 biomolecules, and a very, very small percentage is going to make up ions like different salts and small molecules. Small molecules like salt and ions like potassium. And here are the 4 biomolecules, and you can see proteins make up a larger portion of them than nucleic acids, which is your DNA and RNA. Your energy molecules, lipids, and carbohydrates are going to make up a smaller portion of it. So you need to know for each of the biomolecules the monomers or building blocks that make them up. For carbohydrates, which is what we're going to focus on today, or for these notes, their building blocks are called monosaccharides. Mono again means one, and then saccharide means sugar. And so monosaccharides essentially means single sugars. And what makes a monosaccharide is that it has in its molecular formula carbon, hydrogen, and oxygen atoms in a 1 to 2 to 1 ratio. Okay. So for example, glucose C6H12O6 is a monosaccharide because it has 6 carbons to 12 hydrogens to 6 oxygens, which reduces to a 1 to 2 to 1 ratio. So a carbohydrate is either one or more sugars bonded together. And again, their monomers are monosaccharides. Proteins, their monomers are amino acids. Nucleic acids, their monomers are nucleotides, and lipids, their monomers, well, it's not technically monomers because they're not repeating, but within a lipid molecule, there is glycerol, at least one glycerol, and at least one fatty acid molecule. And we'll go back to these slides when we do this slide when we do the other 3. One thing to understand is within a submolecule, there are what's called functional groups attached to the carbon skeleton, and that's going to determine the chemical properties and reactivities of that particular molecule. So here are the main functional groups. I'm going to go over the ones that are super important right now. So hydroxyl, you're going to want to write there polar and hydrophilic. If there's OH coming off of the carbon skeleton, that is going to make that portion of the molecule hydrophilic. It's going to enable it to dissolve in water. It's going to form hydrogen bonds with water molecules and other polar molecules. And then the opposite of that would be methyl groups. These are nonpolar and hydrophobic, and that's going to be just those hydrogens coming off of the carbon. So when you, like, for example, when I drew this one that's just a chain of carbon and hydrogens, that would be a bunch of methyl groups, and it would be completely nonpolar. Phosphate groups we'll talk about a lot with DNA and RNA, but that's going to make the molecule negatively charged. Amino groups and carboxyl groups are found at the ends of amino acids, which are the building blocks of proteins. And the amino group is polar, and the carboxyl group is both polar, and it's going to be acidic because it can donate a hydrogen. So you need to know how monomers are used to build polymers and how polymers are broken back down into monomers. So inside of cells are constantly working to build new molecules, and they build those molecules out of recycled parts. And those recycled parts come from breaking down polymers that you take in your diet. So you eat other organisms' biomolecules, or you take in other organisms' biomolecules, and you're going to break them down back into, you're going to break those polymers back down into monomers, and you're going to use those monomers to bond together to build polymers inside of your body. So how you build polymers from monomers is by covalently bonding monomers together through a reaction called dehydration synthesis. And this is also called condensation, because when this reaction happens and the bond is formed between two monomers, a water molecule condenses out or drops out, and it's going to be a waste product of the reaction. And so the summary of a dehydration synthesis reaction is monomer A plus monomer B yields product AB. So A and B are now bonded together plus one water molecule as a waste product, and that's why it's called this. Synthesis means to build, dehydrate means to lose a water molecule. And this summary diagram is showing a basic reaction of dehydration synthesis. So here's my two monomers, and this one has a hydroxyl group coming off of the carbon skeleton, and this one has a hydrogen coming off of the carbon skeleton. When they get close enough together, and usually by an enzyme, always by an enzyme, the hydrogen is going to get pulled to this hydroxyl group. A water molecule is going to form because OH plus H is H2O, and that leaves these two carbon skeletons with empty shells, and so they're going to need to bond together to fill in that, to gain those valence electrons to share them to fill their outer shells. And so this red line represents the bond between these two monomers, and here's the water molecule that's dropped out, or right here. And this happens, so now another monomer comes in, and the same thing's going to happen, and now another bond is formed. And this can happen until there's hundreds to thousands of these monomers bonded together. This is showing the dehydration synthesis of sucrose, for the formation of sucrose from two monosaccharides, glucose and fructose. So these are two single sugars, and here's two hydroxyl groups right here. And what happens is an enzyme is going to bring them close enough together where this hydrogen is going to get pulled to this hydroxyl group. And when that happens, a water molecule will form. OH plus H yields H2O. And now I have this empty carbon right here that lost its bond with oxygen. And then I have this oxygen right here that lost its bond with this hydrogen. And so this oxygen is going to get pulled towards this carbon right here and form a covalent bond right here. And this is called a glycosidic link because we're talking about the formation of polysaccharides. Okay, so when we're breaking down polymers, like in digestion reactions, you need to break down your food into smaller components in order to rebuild things or to use them for energy. Then you're going to perform what's called hydrolysis reactions. And this is the exact opposite of a dehydration synthesis reaction. So now I have my sucrose, and another enzyme is going to do this. I know the name of this enzyme is sucrase. And instead of losing a water, you're going to add a water. So water is going to be a reactant. So I have dimer A plus B, I'm sorry, dimer AB, which is the two monomers bonded together. So in this case, AB would be sucrose plus water, and that is going to yield monomer A plus monomer B as the products. And so to summarize what happens, the water molecule, using its polarity, is going to disrupt this bond right here and break it apart. And part of the water molecule is going to come to the to this side, and part of the water molecule is going to come to this side. So if this is the oxygen right here, it can gain a hydrogen and form this OH right here attached to the carbon. And then over here, the OH left from the water is going to bond with this carbon right here and form this hydroxyl group. And so therefore matter is never created nor destroyed. An OH goes to one and an H goes to the other to attach to this oxygen. So now these two monomers have these two hydroxyl groups here made from the water and this oxygen right here. So you are performing an experiment that involves a hydrolysis reaction. So that means to break with water. Hydro means water, lysis means to break. The polymer that you are working with is amylose starch. Starch is a polysaccharide, meaning it's made up of hundreds of monosaccharides bonded together. This specific one, amylose, has 300 monomers of glucose that make it up. How many water molecules will you need to completely hydrolyze the amylose starch polymer? Well, first of all, in this picture right here, I have 1, 2, 3, 4, 5 monosaccharides covalently bonded together. Here's those glycosidic links. And so there's 1, 2, 3, 4 glycosidic links that are needed to bond these monosaccharides together that were formed in dehydration synthesis. And so therefore, if there's 5 molecules, there's 4 bonds between them. So every time you perform a hydrolysis reaction, you need 1 water molecule to break 1 bond. And so you would need 1, 2, 3, 4 water molecules to break these 4 bonds. And so if you have 300 of these sugar molecules, you would therefore have 299 bonds, and therefore you would need 299 water molecules to break them. All right, carbohydrates. So again, the monomers are called monosaccharides. Their formula is carbon, hydrogens, and oxygens in a 1 to 2 to 1 ratio. For example, glucose is C6H12O6. Monosaccharides contain multiple carbonyl and hydroxyl groups. Therefore, they are polar and hydrophilic. Sorry, I'm going to sneeze again, I feel like. They're primarily used as short-term energy storage, but some are used as building blocks in other molecules such as ribose and RNA. And when I say short-term energy storage, that means that they are going to be eventually just completely catabolized, broken apart for energy. But if it's used as a building block in other molecules, it's a structural molecule. It's not going to be broken down for energy. Monosaccharides bond together to form disaccharides. Di means 2 through dehydration synthesis. And then later, disaccharides will bind with other monosaccharides to form trisaccharides, oligosaccharides, and eventually polysaccharides. Disaccharides have similar properties to monosaccharides. They're still polar. They get hydrolyzed into monosaccharides, and then those monosaccharides can be used for energy. Examples of disaccharides would be sucrose and lactose. Sucrose is table sugar, and lactose is milk sugar made in mammals to feed their young. Here's ribose and deoxyribose. They're 5 carbon monosaccharides, so C5H10O5. And although technically deoxyribose has 1 less oxygen. We'll go into that way later. And again, they're not broken down for energy. A lot of monosaccharides are C6, so here's all 6 carbon sugars, or hexoses as they're called. And 2 6 carbon sugars are used to form sucrose, which is table sugar. And that water molecule is lost when this OH and H come together to form it. So check for understanding, how do you build sucrose? So first of all, this is going to be a dehydration synthesis reaction. And I have C6H O6, which is glucose, plus C6H12O6, which is fructose. They have the exact same molecular formula, but the arrangement of the atoms within the structure are slightly different, and that's why they're different molecules, so they have slightly different properties. And then when you perform dehydration synthesis, waste product is going to be 1 water molecule per bond, so we're going to lose a water molecule. And matter is never created nor destroyed, and so you're going to think about that when you're figuring out the molecular formula of sucrose. So there's going to be 6 plus 6 carbons, so there's going to be 12 carbons in sucrose, and then there's going to be some hydrogens and there's going to be some oxygens. How many of each? Well, there are 12 plus 12, 24 hydrogens that come together, but 2 are used in the formation of water, so therefore 24 minus 2 is 22. And then there's 6 plus 6, 12 oxygens, and 1 is used in the formation of water, so 12 minus 1 is 11. So therefore, the molecular formula of table sugar is C12H22O11. Sorry guys, struggle with technology. So here's hydrolysis of sucrose. Here's the enzyme sucrase that is going to perform this reaction, and we'll get into enzymes in another unit. You need to add a water molecule to disrupt this glycosidic link, which again is a type of covalent bond. Polymers are always, the monomers are always bonded together covalently. And water disrupts that bond, and now here's the OH and H that come from the water molecule. So these two monomers are going to be the two products, and they're going to be more polar than sucrose because they now have these extra hydroxyl groups. Oligosaccharides are carbohydrates with few to several monosaccharides bonded together, and they are often not used for energy, but they're used as structural components, often found on the cell membrane, either attached to a receptor protein or to some sort of lipid embedded in the cell membrane, and they often serve as recognition signals or markers for cell communication. For example, what blood type you are, A, B, and O, is dependent on what oligosaccharide chains you have hanging from your cell membrane. So here is, for example, a receptor protein, and here's the oligosaccharide chains coming off of it. And you can see them in orange right here. They're going to determine like what molecules can come into and out of the cell, what cells the cell can attach to. Last but not least, we have polysaccharide, which I don't like how I wrote the definition here, but a polysaccharide is anywhere from hundreds to thousands of monosaccharides bonded together. And you need to know these 4 monosaccharides. These 4 polysaccharides. Starches are polysaccharides that are made and stored in plants. And when a plant performs photosynthesis any photoautotrophic organism, it's going to make monosaccharides. And it'll use those monosaccharides for energy, but if it has an excess amount, it will store its monosaccharides like glucose it makes in photosynthesis by bonding those glucose molecules together to form amylose, or as we call it, starch. And by bonding them together, it's going to make them more compact and allow the cell to not just be filled with monosaccharides. It's going to be more organized storage. Think about like one of those like vacuum seal things that you get to store like winter clothes or something like that. And they're going to be stored in chloroplasts in these little granules called plastids. And then when the plant needs those starches, it can hydrolyze them back into glucose molecules. It could also convert its starches into other polysaccharides, like for example cellulose, if it needs to build more cell walls during cell division. Glycogen is how animals store their glucose. So when our blood sugar level is too high, what we do is we, in our liver, take in the glucose from the blood and bond them together to form glycogen. Glycogen and starch are very similar structurally, but the one difference is glycogen is going to be more highly branched, and therefore you're going to be able to fit more glucose molecules per unit space in glycogen. Cellulose makes up plant cell walls, and chitin makes up exoskeletons in arthropods and arthropods, which are crustaceans and insects, and also fungal cell walls. Cellulose and chitin are different structurally from starch and glycogen in that there isn't many organisms that have enzymes that can hydrolyze the bonds in cellulose and chitin. And so therefore it's super strong structurally and not going to be used for energy. This diagram shows first cellulose. You can see that each monosaccharide monomer in cellulose is flipped upside down. So this one, here's the hydroxyl group, here's this one, here's the hydroxyl group. So they're like isomers of each other, if you learn that in chemistry. And so that's going to create this kind of up-down bonding fashion. And that's what makes it virtually impossible to break. There's only a few microorganisms that have the enzyme cellulase to break it down. And so cellulose is going to be used as the molecular structure to build plant cell walls. Now, if you've heard the term fiber, what fiber is plant cell walls. And you actually can't digest fiber. It goes right through you. Well, why do we need fiber in our diet? Well, the plant cell walls, as they're going through your digestive tract, the cellulose, it kind of like picks up other polar molecules that are stuck in your digestive tract and helps move things along because it's just going to run right through you. So yeah, older people need to eat a lot of cell walls. Here is glycogen versus starch. Here's starch right here. Each of those green things is a single sugar molecule, and here you can see it's just thousands all bonded together. Here is glycogen, and it's going to be more highly branched, and therefore it's going to be more dense, and there's going to be more glucose per unit space. Okay, so in this image right here, it's really important you understand this. When a plant photosynthesizes, it makes sugar molecules, which are these red hexoses. And if it has an excess amount, it can bond them together to form this molecule right here, starch, which is going to store in its plastids. That starch can then, some of it can be used and converted to make cellulose, done by other enzymes inside of the plant cells. And then when a squirrel eats the plant, it's going to get its stored monosaccharides that it could potentially have, stored polysaccharides like starch, and cellulose. The cellulose is going to go right through the squirrel and he's going to poop it out. But the starch, the squirrel's going to be able to use its enzyme amylose to hydrolyze into monosaccharides. And a lot of those monosaccharides will be used for energy for the starch or for the squirrel to go about its daily business, running up trees and just being super cute. And the rest of it, if it gets an excess amount, it will then, after it's hydrolyzed the starch into glucose, in its liver, it'll, through dehydration synthesis, bond those glucose molecules to form glycogen and store the glycogen in its liver and muscle cells. And then if the squirrel, like right now where it's really hot and not a lot of plants are available because it doesn't rain at all, it's going to hydrolyze its stored glycogen into glucose molecules to keep its blood sugar level high, so that way it doesn't die during tough times. Structurally, chitin is going to be really similar to cellulose, except for off of each of its monosaccharides, it's going to have a nitrogen-containing compound or molecule right here. And the exoskeleton of bugs, again, is going to be structurally made of chitin, and it's so strong, it's how we make surgical thread. This is a nice summary chart of all the carbohydrates, monosaccharides, polysaccharides, and everything you need to know about them. So I'm going to stop here with the notes. Make sure you have them filled in when you come to class next. Thank you.